The bulk of the cell membrane is made up of phospholipid molecules. These molecules are arranged in two rows. The molecules within the same row often exchange places with each other. They're constantly moving like a beaded curtain. The hydrophilic heads of the phospholipid molecule is facing outwards, and the hydrophobic tails, they face towards the interior of the phospholipid bilayer. The cholesterol that we see in green here stabilizes the phospholipid molecules so they don't move too much. Membrane protein is shown in blue here. There are many different kinds of membrane proteins. The next component is the carbohydrate chain. There are many carbohydrate chains all along the outer surface of the cell membrane. If a lipid is attached to a carbohydrate chain, we call it a glycolipid. When a protein is attached to a carbohydrate chain, we call the protein a glycoprotein. Now for the functions. Due to its hydrophobic interior, the phospholipid bilayer is very specific in what it can allow to pass through. Only very small polar molecules or hydrophilic molecules are allowed in. However, large polar molecules are not allowed to pass through the phospholipid bilayer. We call this property semipermeability, and it also forms the bulk of the cell membrane. The next component is the membrane protein. There are many different kinds of membrane protein on a cell membrane. They could be a receptor, an enzyme, a channel, a gated channel, a cell adhesion molecule, or a cell identity marker. The next component is the carbohydrate chain. The carbohydrate chain, when attached to a protein or a lipid, they act as identity markers, cell-to-cell identification. And they also serve a protective function because there are so many of them on the surface of the cell membrane. Cholesterol's function is to stabilize the phospholipid bilayer when the temperature is high and the phospholipid molecules tend to move a little too much. What can and cannot pass through the phospholipid bilayer? Here is the phospholipid bilayer in pink. We need to remember two rules before we consider different kinds of molecules. The first rule is the phospholipid bilayer has a hydrophobic interior. The second rule is that the phospholipid molecules are constantly moving and exchanging places, which means very small molecules can make it through. So let's look at these one by one. Here we have oxygen, carbon dioxide, and water. These three molecules are very small, which means they can squeeze through the phospholipid molecules, the space between the phospholipid molecules. So they can pass through very easily. Now let's take a look at some hydrophobic molecules. Lipids are hydrophobic. They don't like water. Water and oil do not mix. So all lipids are hydrophobic. That would include steroids, such as hormones, and fats, which are triglycerides. All of these can easily pass through the phospholipid bilayer because they are hydrophobic. Let's look at ions. Ions are charged atoms, which means they are hydrophilic. Ions are also fairly big. They cannot fit in the spaces between the phospholipid molecules. So they don't satisfy both rules. As a result, ions cannot pass through the phospholipid bilayer. They are big and they are hydrophilic. The next example is glucose, which is a monomer. It cannot pass through the phospholipid bilayer because, like the ions, glucose does not satisfy both rules. It is hydrophilic and it is too big. There are several different kinds of membrane proteins, so let's look at them one by one. The first protein on the extreme left is a receptor protein. You want to think of a receptor protein as a bouncer in a bar. Your bouncer checks your ID before he lets you in. So the receptor protein is like a bouncer. The second protein is an enzyme protein. The third is a channel. The channel is always open. The fourth protein seen here is a gated channel, which means the channel can open or close depending upon the stimulation. The fifth protein is a cell identity marker with a carbohydrate chain attached to it. This allows cells that are similar to identify each other, and then the last protein, cell adhesion molecule, helps these like cells to stick together. We call this CAM, cell adhesion molecule. Membrane transport. There are two kinds of membrane transport. There is passive transport and there is active transport. Passive transport does not use ATP, whereas active transport uses ATP. That's the main difference between the two. So let's write it down here. Passive transport does not use ATP. Active transport uses ATP. Passive transport. Passive transport is mostly the movement of molecules down the gradient, which means they move from a region of high concentration to a region of low concentration. So let's write that down the gradient. There are three kinds of passive transport: simple diffusion, facilitated diffusion, and osmosis. So let's take a look at simple diffusion. This is the movement of molecules down the concentration gradient. So you move from a region of high concentration and molecules move down to a region of low concentration. So now let's take a look at facilitated diffusion. It's very much like simple diffusion, but the only difference is that facilitated diffusion needs a protein channel, which means larger molecules will move through this, polar molecules will move through this channel. In simple diffusion, the movement of molecules are usually very small. So here we have a protein channel for facilitated diffusion. Now let's move to osmosis. Osmosis is the movement of water from a region of high concentration to a region of low concentration of water molecules. So water usually moves from a place where there are fewer solutes to a place where there are more solutes. They move from a region of high concentration of water to a region of low concentration. In other words, water moves towards the salt. Now let's look at active transport. Active transport is different from passive transport in that all the three types of active transport require energy in the form of ATP. There are three main kinds of active transport. They are primary active transport, secondary active transport, and bulk transport. Primary active transport is brought about by a protein pump that is powered by ATP. You need this pump because molecules are moving from a region of low concentration to a region of high concentration. The molecules are moving up the concentration gradient. In secondary active transport, which is very complex, there are two different transports going on at the front end and the back end of the cell. The front end has molecules moving down the gradient, and the back end has molecules moving up the gradient. Bulk transport deals with anything that's very large. It could be a bacteria, a virus, it could be many antibodies being released by a cell. If the molecules are entering the cell, we call it endocytosis, and if molecules are leaving the cell, we call it exocytosis. Bulk transport deals with very large molecules or small molecules packaged together. We need to remember that active transport requires ATP.